the blood and the myeloid tissue by identifying the principal codes of the eyeball and the features of its growth. We'll be tracing the aqueous humor from its site of production to its drainage. We'll be identifying the accessory organs of the eye and their parts. We'll be identifying the lymphoid organs based on their unique specific characteristic features and locating their different areas, zone, and spaces. We'll be identifying the mature blood elements as you will see them on the peripheral smear and also in the bone marrow. And we'll try to identify the different blood elements at different stages of their development. Okay? Microslide projection will be of great help to you if only you're familiar with the slides that are assigned for you. Okay? If this is your first time to see the slides, unless you have a photographic memory, you'll not get much out of it. Okay? Okay, I have this. This is not actually a slide. I took it from Junquera. Because considering the slide, the eye, you can have it even with 4x in a whole field. Okay? Just for introduction, the eyeball has 3 codes. You have the fibrous code. Okay, you have the nervous code as the innermost, and in between you have vascular code. But these 3 codes may not be seen in one single field, the kind of slide that we have. So it's either you see parts of these 3 codes at the posterior part of the eye or the anterior part of the eye. Okay? So for the fibrous code, it is made up of the main sclera, which consists of 5/6 of the whole eye, and 1/6 will be anterior. You have the you have the cornea. Okay, and then for the vascular code, it is made up of 3 components. You have the choroid. Okay, the choroid. That's the main part of that, and the most anterior is your iris. Okay, and the ciliary body. Then of course you have the nervous code, which is mainly the retina. Okay. So for the first slide, some of you might have this slide, but others will have a different slide. No? So with this slide, you'll have the 3 codes. You have the fibrous, which on the posterior part is the sclera, and then you have the vascular code. In this part you have the choroid, and then of course you have the nervous code, which is your retina, which could be seen as having different layers. Okay? So for the sclera, it is homogeneous because it is made up of fibers, either collagen or some elastic fibers. Okay? It is opaque, as compared with the more anterior 1/6 of the fibrous coat, which is the cornea. The cornea is very transparent. Okay? So it is fibrous because it protects the rest of the inner eyeball. Okay? Again, the fibrous is made up of your sclera and the more anterior cornea. Okay? Then of course you have the vascular coat, which is composed of your choroid, ciliary body, and your iris. And the nervous is mainly your retina. Okay? So posteriorly, okay, you have the sclera, and it is the opaque, as I said, compared with the more transparent cornea. And the junction between the 2 is what you call your limbus. Okay? So you have the transition from your opaque part of your sclera. The fibers will degenerate with the sclera here, and this area is important because this will be the area for the drainage of the fluid where you will have your aqueous humor. Okay, we'll go to that later. So again, this is the area of the limbus, or characteristically is the junction between your sclera and the cornea. So for the cornea, which is the more transparent, a vascular part of the fibrous coat, Okay? It is made up of 5 layers. Okay? The outermost layer is the epithelium. The innermost layer is also an epithelium, but this time it is composed of single layer of squamous cell. This time, in as much as you have the fluid here, and it is a serous membrane, you call this time endothelium. It is also an epithelium. Okay? So, hence, this is your outer epithelium, this is your inner epithelium, but specifically we call it your endothelium. Okay? The epithelial cells of your first layer will be resting on the basement membrane, and you call it your Bowman's membrane. The epithelial cells of your endothelium will also be resting on the basement membrane that will comprise your posterior limiting membrane or the desciments. And the main part, the thickest part of your cornea is your stroma or substantia propria. Okay? How to remember? Outer layer, epithelium. Inner layer, endothelium. In as much as they are epithelial cells, they'll be resting on basement membrane. How to remember anti-epithelium, anterior limiting membrane? Bowman's, as against desciments for the posterior limiting membrane. So, outer, letter B, Bowman's. And then the posterior, the letter D, desciments. Okay? Then mainly you have the stroma. Okay? The outermost layer of the cornea is very sensitive, no? So it's highly innervated by fibers. Hence, a little neck to your cornea will give you a lot of pain. So for those who have your contact lens, diba? The cornea is very, very sensitive. And you will see that it is avascular. No blood vessels in any of the layers. Okay? Because the nutrition here is by diffusion from here and also from this on this side. So you remember cornea, epithelium, the first layer, endothelium, the fifth layer, and the third layer you have the stroma, and the second and the fourth layer will have the basement membrane. So anterior and posterior, Bowman's and desciments. Okay? Can you remember them? Okay. Okay, for the vascular coat, you call it also uveal tract because this coat is very vascular and that will give you a violaceous color. Uve means violet. Oh. Diba? Uve is violet. Uveal tract, very vascular. Okay? So as I said earlier, it has 3 components. You have the choroid, diba? Ciliary body, and your iris. But for the more posterior, the main part is your choroid. It's located in the posterior compartment of the eyeball. It is further subdivided into suprachoroidal layer, choriocapillary layer, and your Brooks membrane, another membrane. Ano yung kanina? Bowman's, the outer. The inner is your desciment. This time another B, Brooks. Okay. So, we have said earlier that choroid is very vascular. Okay?
Right.
And you will see the blood vessels or the capillaries will be seen in choriocapillary layer, where you have the capillaries of your choroid. So choriocapillary layer is the main layer of your choroid. Okay? Above it is your suprachoroidal layer or lamina. And then of course, you have the avascular, the acellular layer, which is your Brooks membrane. Pag membrane, lagi medyo acellular. Okay? It's either you have the basement membrane or the cellular part. Okay? So Brooks membrane is for the choroid, and the desciments and the Bowman's for your what organ? Cornea. Okay? So given on low power magnification, no, your choroid will have a lot of melanocytes. melanin pigments. Okay? Especially in the area of suprachoroidal layer or lamina. So it's very black. The suprachoroidal layer is will have a lot of melanin pigments. Compared with the choriocapillary layer, well, you have here the cross-section of your capillaries. So a little pinkish, open spaces there. Then the clear area is your Bruch's membrane. So your Bruch's membrane is intimately related to the first layer of your retina, which is your pigment epithelium. Okay? There, suprachoroidal layer or lamina, choriocapillary layer, and then Bruch's membrane. Okay. So the second component of your vascular coat or your uveal tract, you have your ciliary body, which is composed of the main part of your ciliary body is formed by your ciliary muscle. Okay? It is a smooth muscle. Okay? So it is by autonomic innervation. Okay? So aside from the main ciliary muscles, you have finger-like projections from the main body, which is your ciliary processes. The ciliary processes will have a lot of fenestrated capillaries. Okay? And it is at this part that the aqueous humor will be produced. So aqueous humor is ultrafiltrate of your serum that will happen in the fenestrated capillaries that you'll have in your ciliary processes. And then attached to your ciliary body, you'll have your ciliary sonnular fibers that will support or anchor your lens into the mid part of your ciliary body. So they are thin fibrils that will anchor the lens into the ciliary body, the midpoint of the ciliary body. So they will the fibers of fibrillar structures that will enclose your lens. Okay. So with the contraction of ciliary muscles, that will, what happens to the sonnular fibers? With the contraction, okay, that will relax or contract the fibers. It will, it's always a reverse. You contract this, you relax the fibers. When you relax this, what will happen? The fibers are going this way, and this way. So that when you relax, you will lessen the pressure onto the thickness of your lens. Hence, with the contraction of this, you have thickening of your lens. Okay? Contraction of ciliary muscle, relax the sonnular fibers, hence you'll have your thickening of the lens. Okay. Between the ciliary body, at the junction of the ciliary body, and also with the iris, together with the cornea, you have there a space where you have your canal of Schlem. Okay, so this is space which serves as the drainage of your aqueous humor. Okay, so from the ciliary processes, the production of this aqueous humor, it will go to your posterior chamber, pass through the pupil in between the iris, go on to your anterior chamber, then drain into your canal of Schlem, eventually find its way to your episcleral veins. Okay? So the third component of your uveal tract is your iris. Okay? It is radially disposed in the eye, and it is at times it is abutting your lens. Okay? So you have it projecting from the superior end of your ciliary body. Okay? So the anterior surface of your iris has no covering. compared with the posterior layer, okay, which will have a lot of melanocytes. Well, all over the iris will have melanin pigments, but you'll have more on the posterior surface, okay? Notably, you'll have the muscles that will move the iris. No? You have the dilator pupillae. No? This is the constrictor pupillae. Sorry. Reverse. Sorry. So this is the constrictor pupillae and the dilator pupillae. The constrictor pupillae is towards the pupil. It is circularly disposed, like that. Okay, you have to reverse that. You have to reverse the label, okay? Okay. So you have here the sphincter pupillae because it is circularly disposed around the pupil. So that when this sphincter pupillae contract, it will constrict the pupil. Okay? The dilator pupillae is thinner and it extends along the length, more or less, of the iris, but more prominent to the midline and towards this. So it's very thin. Okay? So you just expect that there are some few muscle fibers here. Okay? So dilator, sphincter pupillae and dilator pupillae here along this line. Very thin compared with the thick sphincter pupillae. Okay? And then of course between the two ends of your iris, you have there your pupil. Okay? Which is the That's a continuation of your posterior chamber with a anterior chamber via your pupil. This is another slide, and you can see what kind of slide you have. Anyway, one thing that you have to remember in the slide is if you see this dense red thing there, on one side, is that of the eyeball. That is the lens. Because most often than not, we cannot have the whole eyeball in field. So most often than not, so the reference point is the lens. And the black thing there is your iris. And then of course, until you do that is your what? Cornea. And then over there, okay, and the muscle is your ciliary muscle there, and then ciliary processes, okay? So here you have the anterior chamber and the posterior chamber. Okay, so with the uveal tract, these are the things that you have to identify, okay? Okay, your ciliary body, ciliary muscle, processes, okay? And of course, here you have a good sonular fibers. Okay. Then of course, on the iris, no, we have the constrictor pupillae, dilator pupillae, and then anterior chamber, posterior chamber, and the fluid that will be contained in this chamber is your aqueous humor. Okay. You may be asked what is found in the pointed space. Okay. Okay, now we go to the third coat, which is your nervous coat. So this is the posterior compartment of the eyeball. See, again, you have the sclera, the choroid, but the pigment epithelium is attached to your choroid here. And the main part of the retina is usually detached in this area. Because the pigment epithelium, which is the first layer of your retina, is derived from your optic cup. Okay? So these two will have a different site of origin, okay? So how will you know that you're looking at the retina? You have layerings, okay? How will you remember? Remember that the retina will have 10 layers. Have you memorized them? Why not? Okay. What are the key layers that you have to remember? There are 3 main layers that you have to remember. Huh? The layers that will have the cell bodies of neurons. Okay, you have the 4th, the 6th, and the 8th. You have the outer nuclear layer, inner nuclear, and the layer of your ganglion cells. 4, 6, 8. Kaya nyo tandaan yun? Of course. Okay, so in this layer you have your cell bodies. Cell bodies of your rods and cones cells. Cell bodies of mainly your bipolar cells. And here, cell bodies of your ganglion cells. If you have cell bodies, it will have its corresponding dendrites and axons. Okay? So, the axons of your cell bodies of your rods and cones will synapse with the dendrite of your, mainly your bipolar cells of your inner nuclear layer. The synapses will happen, okay, you have your outer plexiform layer. Connections, di ba? Plexus. Okay? And the axons of your inner nuclear layer, which is composed mainly of your bipolar, okay, of your bipolar cells, and okay, so the axons of your bipolar cells and the dendrites of your ganglion cells will give you the inner plexiform layer. Okay, so you have 5 layers right away. Okay, so what's next? Okay, so you have earlier the connections of your axons of ganglion cells with dendrites of your ganglion cells with the axons of your bipolar of your nuclear layer, as we said before. This time, the axons of your ganglion cells will make up a special layer. This is your axons of ganglion cells. These fibers, these axons will eventually exit the eyeball by as a single unit, no? That will be your optic nerve. So if you're asked if the optic nerve is tight, what does this represent? The answer is axons of ganglion cells of the retina. Okay, so the cranial nerve 2 or your optic nerve are the axons of the layers that you have in the retina, specifically ganglion cells. Okay? Then of course, you have axons of ganglion cells. We haven't mentioned yet the dendrites of your rods and cones, because in the outer nuclear layer, you have the cell bodies of rods and cones. So you have actually the layers of rods and cones. These are specialized photoreceptors, which are actually dendrites of your rods and cone cells. Okay? Then of course you have the outer limiting membrane again. If you have outer limiting membrane after the layer of rods and cones, you have the inner limiting membrane after the axons. Because remember, your layer of rods and cones dendrites. After the dendrites, you have the outer limiting membrane. After the axons of ganglion cells, you have the inner limiting membrane. Okay? So you have there your 9 layers. And then of course, you have your pigment epithelium, which is not shown here. The slide is that of, the slide talaga ng retina, labeled as the retina. The slide of the retina will not have the pigment epithelium. So you have to appreciate the 10 layers in the previous slide, where you had the 3 coats. Okay? Okay? Again, outer nuclear layer will have the cell bodies of rods and cones. Your inner nuclear layer will have the cell bodies of bipolar cells. But aside from bipolar cells, you have other cells there. You have the horizontal cells, you have the amacrine cells, plus the cell bodies of your Mueller cell. So remember that this layer will have more than one type of neuron. Okay. And then of course you have your ganglion cells. The thickness of your ganglion cells will differ on the location of the retina. The more central the retina is, the more ganglion cells you'll have. The more peripheral the retina is, no? The outer part, no? You'll have a single layer of your ganglion cells. But in some areas, you have very thick ganglion cell layer. Okay, so you have here, So, again, this is your sclera. This is your choroid. Okay? This is your pigment epithelium. And then the rest are the 9 layers of the retina. Okay? The two-tone part there is your layer of rods and cones. It appears two-tone because you have the part where you have only the rods, the part where you have the rods and the cones. Yeah, two-tone. Okay? Then, so after that is the dark area, which is your 4th layer, your outer nuclear layer. The lighter area, which is your outer plexiform layer. Then you have the 6th layer, which is your inner nuclear layer, where you have the bipolar, horizontal, amacrine, and Mueller cells. Ang dami. But mainly bipolar cells. Okay? So after that, again, a light area. Again, you have your inner plexiform layer. And this time, the next is ganglion cell layer, or cell bodies of your ganglion cells. So here, we have only 1 or 2 layers of your ganglion cells. But in this area, you have it thick. Towards this, you have all the thinner. Okay? So depending on the layer, on the part of the retina, you could either have a thin part for your ganglion cells, but you may encounter this thicker part. Okay? So don't memorize which is the thickest. Okay? So your point of reference is your rods and cones. Okay? Because that is the one that will be outermost. Okay? Okay, sorry. So after your ganglion cell layer, so you have here your, okay? So you have the axons of your ganglion cells. And then, well, not shown ordinarily, not shown will be the inner, the outer limiting membrane and the inner limiting membrane as the 10th layer. Okay? You can remember? Okay, so this is the other slide that you may have, no? So you have here your rods and cones layer. You have the outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer. You have here your ganglion cell layer, and this part is your axons of your ganglion cells, and there may be here ito. This is the remnant of your inner limiting membrane. The inner limiting membrane will separate the retina from your vitreous chamber. Okay? Yeah. Okay. Just to recap, for the course of the eyeball, you have in the posterior compartment of the eye, you have the sclera as your fibrous coat, the choroid for your vascular coat, and then of course you have the retina. So with this field, you have the 3 coats. But ordinarily, this is seen in your posterior compartment of the eye. Okay? And then for the more anterior part, you'll have the other parts of your fibrous coat, which is your cornea. For the vascular coat, you have your ciliary body, and then the iris. And then, well, a nervous coat is stop at the area where you don't have the nervous tissue, where you have your ora serrata. Hanggang dito lang yung ora serrata. Okay. So now we go to accessory organs of the eye. You have the eyelid as the covering. Okay. So with this is your cornea. Remember? Okay. You have the cornea. Okay. And then of course you have the eyelid. Okay. In the eyelid, it is covered by the skin. The skin is loose, no fat cells. No. And if you have the skin, you have the associated hair follicle. Okay, the hair follicle. But for the inner part of your eyelid, okay, there will be a transition of your stratified squamous epithelium, keratinized, to your stratified columnar epithelium here, which will be your conjunctiva. Okay? So, in as much as this part is covering, upon the inner covering of your eyelid, this is your palpebral conjunctiva. Okay? This is another slide of the slide number. Yeah. So again, the skin, you have for the exit of your hair follicle, and then the transition here, and then this area will be your palpebral conjunctiva. You'll be able to move your eyelid. Okay? You can close and open the eye aperture or the ocular aperture or the opening of your eye by means of your contraction of your orbicularis oculi. So the muscular part here is made up of skeletal muscles, your orbicularis oculi. Okay, they are circularly disposed, no, around the orbit. Yeah. So this represents the cross section of your skeletal muscles. So bundles and bundles of your orbicularis oculi. And associated to the inner lining of your eyelid will be your large sebaceous gland, what you call your tarsal glands. And these tarsal glands will be embedded in a dense fibroelastic connective tissue called your tarsus or tarsal plate. So these are large sebaceous glands or tarsal glands. The other name for that is your meibomian gland. High magnification, again, you have the skin, the palpebral conjunctiva, you have the large sebaceous gland as your tarsal gland or meibomian gland, and this will be embedded in your dense fibroelastic connective tissue. That forms the supporting structure, kind of dense. In fact, it's just like as if you have a cartilage in your eyelid, but it's more of dense connective tissue. That's why you can invert the eyelid. Anybody who would like to demonstrate? Well, you can invert the eyelid as if you have there your the tarsal plate. That's what you call your tarsal plate. Okay? Anybody who would like to demonstrate? You're inverting that actually the tarsal plate. Okay. Okay, so in as much as you have the skin covering your eyelid, hence you have the associated hair follicle. Then of course, if you have the hair follicle, there will be an associated sebaceous gland, there will be the associated sweat gland. The sebaceous gland that you'll have in the hair follicle of the eyelid are kind of small, and they are what you call your sebaceous gland of Say's. Okay, then the sweat gland that you have for the hair follicle of the eyelid is that of apocrine type, okay, of sweat gland. You call that your glands of mole. Well, here, I think this is the sebaceous gland. Yeah, because most often that is really associated with the hair follicle, diba? Well, for the sweat gland, well, I think this, I'm not very sure if this is your gland of mole. Okay? But one thing, you have here your gland of Say's. Okay? And you have there your hair follicle. See, you have here your sebaceous gland of Say's. There. I think this is your, gland of mole. Again, there's another hair follicle there. You have more hair follicle towards the lateral, di ba? You have sparse hair follicle in the middle half. Then you have more and more of that as you go laterally, di ba? Okay, so for the eyelid, you have to have these structures. Have to have them. Orbicularis oculi, tarsal gland, tarsal plate, palpebral conjunctiva, hair follicle, gland of Say's, gland of mole. Okay? You have to know this structure for the eyelid. Okay. Okay. So I mentioned earlier that for the inner surface of the eyelid, you had the conjunctiva, and you have that as your palpebral conjunctiva. It is a mucosal layer that covers the inner surface of the eyelid, and also it will cover the exposed part of your sclera. Okay, which is the sclera of your eye? The white, because it is opaque. The clear one is your cornea. Okay, so it is covered, the white part is covered by your bulbar conjunctiva. You see in the ICS physical exam, I will mention pinkish palpebral conjunctiva. Diba? Anecteric sclera, but actually that will be the area where you have your bulbar conjunctiva. Okay? And then of course the gland that's associated with the eye is your lacrimal gland. It is compound tubuloacinar or tubuloalveolar gland. It is located superolaterally. Okay, so if we have the eye, the enucleated eye, if you see the lacrimal gland, the superolateral, that could give you the laterality of the eye. Diba? So it is compound tubuloalveolar or tubuloacinar. It is likened to a parotid gland. Tignan nyo, parang parotid gland siya. You have serous gland, serous secretory cells, okay, with granules. So it is glandular, okay. Okay. Do you have any question with the eye and the related structures? Now we go to lymphoid tissue. What's so characteristic about lymphoid tissue? You have the preponderance of the lymphocytes in this organ. The lymphocytes could come as a group or really dispersed, especially in the submucosa of gastrointestinal tract and also in your respiratory tract. Okay, so they could come as a capsulated organ or non-capsulated structure. Okay, so you may have your lymphoid tissue as a group. You may have it as a single solitary lymphoid nodule, as in this, and of course, or an aggregated ones, as in your tonsils, Peyer's patches, and your appendix. The capsulated organs include your spleen, lymph node, and your thymus. Okay, spleen, lymph node, and your thymus. Okay, so there are specific structures that will tell you what organ is the lymph node. Okay, so the first organ is your lymph node. It is an encapsulated lymphoid organ. Okay, so what's so characteristic about the lymph node? Medullary cords, as you have in your quiz. Okay, if you have medullary cords, it is located in the medulla. If you have medulla, you have cortex. So you have the three areas of your lymph node. So you have the outer cortex, the inner medulla, and in between you have the paracortex. So within the cortex, you have there your lymph nodules. Okay? In the medulla, you have there your medullary cords, and the spaces will be your medullary sinus. Okay? And between the medulla and the cortex, you have there your paracortex. It is characteristically that part where you'll have your high endothelial venules, as what was in the exam. Okay, what specific type of lymphocytes do you have in your paracortex? B, T, or what? And K. By the way, we
None.
cannot differentiate the different types of lymphocytes in the kind of slice that we have. Okay? By the way. Okay, for a higher, okay, so I proceed. We have the cortex, medulla, and the paracortex. Okay? So we usually bracket it. Make a bracket. And then for higher magnification, so given a lymph node, it is ovoid. One side is convex, the other side is concave. It is on the concave part where you'll have your efferent vessels, lymphatic vessels, the one that will get out. On the convex portion will enter the numerous afferent lymphatic vessels. Okay? Okay, so you'll have here the afferent vessel, lymphatic vessel. But on the other end, you'll have the hilum, which is the concave part, where you'll have the efferent ones, vessel. Okay? So yon again, medulla, paracortex, and your cortex. And within the cortex, you'll find there your lymphoid nodules. And the lymphoid nodules will have a clear area, which you will call as your germinal center or germinative center. Okay. Okay. So lymph node is encapsulated. It is dense fibrous connective tissue and supported by reticular fibers. Okay. So you have there your capsule. Just beneath the capsule is a space, your subcapsular sinus. If it is a sinus, it's a space. Okay? Subcapsular sinus, just beneath the capsule. So, if you have afferent lymph vessels here, the lymph will enter through this area and find its way to your subcapsular sinus. And it will go to your cortical sinus or radial sinus, which will be the area where you'll have the space on either side of the trabecula. Trabecula is an extension of your capsule into the inner parenchyma. Okay, so you have here your trabecula. The space on either side is, some authors will call it trabecular sinus. So the other name is cortical sinus or radial sinus. Okay, and eventually it will bring the the lymphatic fluid, the lymph, into the medullary sinus. Okay, for the filtration of lymph, and from the medullary sinus, it will have to find its way to the efferent vessel and live via your hilum. Again, your medullary sinus is a space between your medullary cords. Cords. These are cord-like sheets of lymphoid tissues, branching cords. Okay. Okay. This is another slide that we have. Okay. We may use this type of slide or the previous one. Okay? Again, you have the cortex, the medulla, the paracortex, the capsule, the subcapsular sinus, and you have there your trabecula, and either side your cortical or radial sinus. Okay? And then for the lymphatic nodule or lymphoid nodule, okay, and then you have the lighter area where you have your germinal center, where you'll have the maturation of the T lymphocytes. Okay. For the medulla part, you have medullary cords, medullary sinuses. Okay. Again, for medullary cords, mainly your B lymphocytes. Your paracortex will have your T lymphocytes. High power magnification, lymphatic nodule, capsule, subcapsular sinus, cortical sinus, trabecula. Alam niyo na to. You'll be able to remember. Okay. Well, I had to connect the two, so that I'll have the whole field. Okay. So you have the convex portion, the concave portion. Here is your efferent lymphatic vessel. Okay? So these are the structures that you have to remember about the lymph node. We have the capsule, the cortex area, the medullary area, and then of course the paracortex, lymphoid nodules, germinal center or germinative center, and then of course, paulit-ulit na ako, medullary cord, medullary sinus. Okay? So again, you have the hilum.
Ang dami pang organ nito, di ba?
Okay? So these are the things that you have to know about lymph node. These are other available slides that we have, but this is, I think, animal, yeah.
Animalistic.
Okay. Any question? Identify the organ. Hmm? You have, seems like there is lobation. Do you have lobes in the lymph node? Wala, di ba? So what's this? You have your connective tissue in between the lobe, but actually incomplete lobation, surrounded by dense connective tissue, and you have there the capsule. So it's one of the capsule, encapsulated lymphoid organ. And this is your, what's the organ? What's the organ? What's that? For each lobe you have the outer cortex and the inner medulla. Oh, what's that? The thymus. Ay, hindi naman. Basta lobated na ganyan thymus. Sa gilid, mas maitim. Okay? So, thymus. Yan. So, this is the other slide. Lobated again. Oh, yan. Higher magnification. For each lobe, you have the cortex and the medulla. But you don't have medullary cord. What do you have to know about thymus?
Sabi, inom lang ako ng water.
It's a primary lymphatic organ. What else? Thymus or T lymphocytes. So it's the area for final differentiation and maturation of your T lymphocytes. Okay, so it is biggest at birth and begin to decline in size and further involute after puberty. Okay, but it will continue its function to produce your lymphocytes thereafter. And one thing that you have to remember is it is derived from two germ layer. Mesoderm, because your lymphocytes, bone marrow is from mesoderm, and then your epithelial cells here are from your endoderm, specifically your third and fourth pharyngeal pouches. Endoderm yun, okay? Yeah. So for each lobe or lobule, you will have its cortex, the darker part, and the lighter area, which is your medulla. Okay? Onto the medulla, you will see there your thymic or Hassell's corpus cells. Okay? This aggregation of your epithelial cells, epithelial reticular cells of the thymus. Okay? There. The other slides that we have. The first slide that we have shown. Okay? You have a lot. LPO, even under LPO, you'll be able to see. Diba? Hassell's corpus cells. Then higher magnification. The older the individual is, the more adipose tissue or connective tissues you'll have for your thymus.
Blair Witch Project.
Okay. The next is your spleen. Do you have lobation?
No.
Huh? Wala. Okay. Do you have lymphoid nodule?
Yes.
Do
No.
you have lymphoid nodule in spleen?
No pala.
You have. Because the white part is considered a lymphoid nodule. Sabi sa quiz eh. That's why spleen kanina is wrong. The white part is considered a lymphoid nodule. Diba? Okay? Again, it will have its capsule. The capsule will send extension into the deeper structure. You have the, again, you have the word trabecula. And then you have the white area, which represents the lymphoid nodule, which is your white part. And then in between the white part, you will have there your red part. Okay? So what do you have in the white part? Okay? But of course. Okay, so you have here your white part, and you, well, for each white part, you'll have your central artery. Ito, central artery. There. Okay? Okay, so you have the central artery. Your central artery derived from what artery? It is a branch of your splenic artery. From your central artery, it will get out of the white part to become your, ano yun? What artery? Peninsular artery. Di ba?
Bawat umaga daw nag-aano siya.
Sino?
Maganda yun sa
Okay. Again, you have the lymphoid nodule. And for it, you will have eccentrically located of the central adhesion towards the side, you have the central artery. This one is longitudinal cut. But here, it is cross-section. Outside of the white pulp, you may have blood vessel, and, well, if it happens to be in the area where you have the trabecula, you call that your trabecular artery. Here, you have the trabecula around it, but it's a cross-section. Okay? You cannot diagnose, you cannot identify that organ as a spleen if you don't see the white part and the red part, the white part with the central artery. You cannot identify the thymus without identifying the access corpuscles and its lobation. Okay? Those are specific features. Okay? And surrounding the central artery, you have condensation of lymphoid tissue which is around it. So peri-arterial lymphoid sheaths, pulse as well, because in the exam. Ito yung pulse, okay? The red is your central artery. The lymphoid tissue around it is your pulse. Okay? In between the white pulp, you'll have cords of lymphoid tissue. You call that splenic cord or your Billroth cord. Rod-like sheets of lymphoid tissue. Okay? And in between your splenic cord is again a sinus. You have there as your splenic sinus. Okay? Pag sinus space. Okay? So it may be asked, identify pointed space. Okay? Identify pointed structure, you have that your splenic cord. Between cords, you have the sinus. Okay? So you have the cords here, and then of course you have the sinus there. Okay? Then the tissues, lymphoid tissue surrounding the lymphoid. Now, this is what you call your marginal zone. Yeah, marginal zone. Okay, again, the white pulp, the red pulp, central artery, the pulse. Okay, take note that you have here is your splenic sinus, splenic cords. No? So your splenic sinus represents the lumen of your capillary. Okay? So, if you have the capillary, it is lined by endothelial cells. And the specific name for the endothelial cells of your of your splenic sinus. You call that your stave cell. Okay? If you have here, you have stave cell. Yan, gilid na yan. Okay? Rod-like, mapahaba. Okay? Stave cell. Okay? So, in between stave cell, will insinuate the T lymphocytes for its recirculation. Diyan dumadaan ang mature lymphocytes going back to the circulation. Okay? Okay, another slide that we have. Okay, you have here your biliary cords. Cords. Okay, makikita mo yung cords talaga. But here you have your splenic sinus, and this is lined by your endothelial cells there. And these are your stave cells. Okay, so from the capsule, there will be extension of the capsule into the deeper part of the spleen by means of your trabecula. And if you find a big vein along the trabecula, you call that your trabecular vein. Trabecular vein. It's a vein, it's thin-walled, and it will contain, ordinarily it will contain blood. Red blood cells there. Okay? So this is another slide that we have. Well, characteristically, it has capsule. We have the white pulp, the red pulp. So it's not that red. So, okay. Again, capsule, white pulp, the red. Hindi nga lang masyadong maganda. But we prefer the other slide. Okay? Anyway, we go to your mucosa-associated lymphatic node, tissue. MOT. You can have gulf, bulk, bronchus-associated lymphoid tissue, gulf, as gut, referring to gastrointestinal tract. Okay, so mucosa-associated. You have the lining of the wall. Here you have the intestinal tract, the intestine. So mucosa-associated lymphatic node. So you find it in the mucosa layer, in between your epithelial lining. Okay? So you have the lining epithelium here. Okay? Then you have here. Either as a solitary or in groups. Okay? So for each lymphoid nodule, again, you have your terminal center. Okay? Solitary nodule. Solitary. You'll have more of this solitary nodule in the colon. No? You can't miss it in the colon. Okay? Okay? In the jejunum and towards the terminal part of the jejunum, you could either have the solitary one or you may have the start of your Peyer's patches. So you have here now the aggregated lymphatic nodules. Here you have the solitary. Okay? This is the start of your Peyer's patches. So Peyer's patches is not exclusive of your ileum. You may have in the terminal part of the of jejunum, you may have them also. Okay? Yan. Aggregated. There's no intervening tissue that you don't have the lymphocytes, okay? This is the slide that we have. Every dot there is a lymphocyte. Okay? This is the lining epithelium, simple columnar epithelium. This is the lining epithelium, and we have no mucosa layer there. Okay? What are M cells?
Dome shape.
In the area where you have the lymphoid tissue, you may see M cells. What are M cells? Remember, it's associated with the lining epithelium. What's the lining epithelium of your intestine? Huh? Simple columnar epithelium. But for your M cells, no? For example here, no? Okay? In between the columnar cells, you may find dome-shaped cell, epithelial cells, no? What's the function of M cells?
Antigen catching.
What's the function of M cells?